So physiology of bacteria, we want to know what is the chemical components in the bacterial cell. And also we want to know when the bacteria grows normally, what type of condition we should provide, like what nutrients it require to grow. And we need to provide the nutrients. Then, except for nutrients, there must be some other factors required, such as the temperature, the proper pH, and also the proper osmotic pressure. So that's what we are going to learn today. After we learn this, we would be able to design the proper condition to culture bacteria in the lab. So that's one reason we need to learn the physiology. We want to culture the bacteria well in the lab. Like in the future, some of you will be doctors in a hospital. And after you collect the specimen from your patient, you will isolate the pathogen from the specimen. And you need to get pure culture in the lab and further study the property of the pathogen so that you can find the proper antibiotic and the proper method to cure your patient. Okay, first we want to know the chemical components in bacterial cells. And here, and you can see that within the bacterial cell, okay, over 70% of the total weight is just water. It's not surprising, right? Because even in human cells, we have about 70% water in our cell. Okay, and water is essential for all type of life, and because all the other molecules, no matter it's big molecule or small molecules, they need to dissolve in water solution, and all the chemical reactions happen in water solution. So it's very important medium for life. And except for water, there are macromolecules here, not macromolecules. So that's the big molecules such as protein molecules, polysaccharides, the sugar molecules, such as the peptide glycan, it contains polysaccharides, and also nucleic acids that include both the DNA and RNA in the cell. And also we have lipids. So that's the major component of cell membrane, the phospholipids. We also have some inorganic salts like potassium, sodium, magnesium, calcium, all these inorganic salts. They are important because they can help to maintain the intracellular osmotic pressure, and also some inorganic salts are important for the enzyme activity. So the enzyme needs to have the iron in the active site so that they can catalyze the reaction. So these are important chemical components in bacterial cell. Okay. And talking about bacteria and other form of life, we want to know how they get energy. Do they get energy directly from either the sunlight or chemical reaction, or they need to consume organic material to get their energy? And based on this, organisms can be divided into two major groups, autotroph and heterotroph. Autotroph means that the organism is able to synthesize organic components with simple inorganic molecules, such as carbon dioxide. An example of this group include like the plants and algae in water, and in some bacteria. Some bacteria can actually use sunlight to produce their sugar molecules. Okay? So that's autotroph. Autotroph can be further divided into two groups. One is phototroph or photoautotroph, such as the green plant. They can derive energy from the sunlight, or some extreme conditions, there are microorganisms, they can use chemotroph. So they derive energy from chemical reactions so that they can utilize that small amount of reaction to survive and support the ecosystem, such as the microorganism in the black chimney in the sea. So because of the chemical reaction, some bacteria can survive in that environment. While heterotroph means that they require organic compounds as a carbon source. This include, well, fungi, some plants, animals, and some bacteria. And we humans belong to heterotroph, right? And we have to eat 3 meals a day so that we can get organic compounds and finally we get our energy. And talking about here, we are talking about bacteria, and you can see that, well, some bacteria belong to autotroph, some belong to heterotroph. But the bacteria we are talking about in medical microbiology, they cause infection in human body. They live in our body, so they belong to heterotroph. Okay, all the pathogens we are talking about belong to heterotroph part. Okay, next we want to know the growth requirements for bacterial growth. So that's important information about physiology. If you want to culture bacteria in the lab, what type of condition you need to provide so that the bacteria can grow? The first one is, well, of course, nutrients. And yeah, they have their material in the cell. You should provide the essential nutrients for their growth. And that's not enough. You should also provide proper physical chemical condition, like proper temperature, proper pH. That indicates the hydrogen ion concentration. And also proper oxygen level. The higher the better? Probably not, right? Some bacteria prefer higher oxygen concentration, while some they prefer lower or even no oxygen condition. And also osmotic pressure. Last time we talked about that bacterial cells have the cell wall, and the cell wall could provide some osmotic protection. But that does not mean that the bacterial cell can survive in any condition, in any osmotic pressure. For example, if in your kitchen you have a jar of honey, okay, the osmotic pressure in honey is very high. So bacteria in honey cannot replicate. They will dry out. Okay. So that's osmotic pressure. Okay, first we talk about the nutrients. These are the essential nutrients required for bacterial growth. Of course, water. Water is essential for all forms of life, and including human, okay? And in our life, in everyday life, sometimes if you want to prevent bacterial replication or bacterial contamination, you can just remove the water. Can anyone give some example? Remove the water, you can keep bacteria out of your life. For example, okay, after the class, you go home and then you have your laundry, wash your clothes. You want to dry your clothes immediately, right? You don't want to keep your clothes wet for a long time, because otherwise the bacteria will grow on it. It's not good. Do you have a dryer in your apartment? No? They just use the sunlight to dry it? It's popular in China, and the dryer is not popular yet, but more and more families are now using dryers. I like dryers, even though I don't have one right now. Okay, so let's remove the water. And yeah, you can have other examples by removing the water to keep it dry so that bacteria don't grow on it. And also carbon source. Carbon. When we talk about carbon source, we are talking about the molecules which can provide energy, to produce energy, like different type of sugars, starch, sugar, fructose, glucose, sucrose. Those are all carbon sources. Does anyone know the song Sugar by the group Maroon 5? No? We can play it later during the break. And nitrogen source. Nitrogen source is so important because in our cells, the important molecules such as protein molecules, in amino acids, there's amine group, right? A nucleic acid, there is nitrogen base, right? So both contain the nitrogen element. You need to provide nitrogen source. And also minerals, they are required for the activity of enzymes, such as the magnesium, calcium, and also others like potassium and sodium. And you also need to provide growth factors. So the bacteria need those growth factors for their growth, but they cannot make it by themselves. For example, some essential amino acids, purine, pyrimidine, and also some vitamins. And we call these molecules the growth factors. So after we know these essential nutrients, we can mix them together to culture bacteria. So when we mix them together, we form some artificial mixture. And this artificial mixture, we call it medium to culture bacteria. It looks like this medium. Anyone know this medium? This is my beer. So it looks like a beer. The color, most of the medium look like this yellowish color. Okay, except for these essential nutrients, we need to also provide the proper physical or chemical condition to support the bacterial growth. The most important one is temperature. For each specific species, you need to provide the proper temperature. Like some bacteria, actually, they don't prefer a high temperature. They like a low temperature. This group, we call them psychrophilic forms. This word, psychrophilic, means they like low temperature. So they grow best at low temperature, like 15 to 20 degrees. One example of this group is the Listeria monocytogenes. Has anyone heard of this name? Listeria monocytogenes. Listeria monocytogenes is a gram-positive bacteria. It can actually grow in your fridge at 4 degrees, in the fridge, 4 degrees. So if you go out to have some dinner in a restaurant and you take some leftover to home and leave it in fridge, don't leave it there for a long time, because still in the fridge, some bacteria can grow on the food, on the leftover. And Listeria monocytogen is one of the major food poison bacterium we encounter a lot. Okay, the second group is called mesophilic form. The name here, mesophilic, means they prefer some temperature in the middle. So that's the temperature between 20 to 45 degrees. Okay, and the human body temperature is in this range. Most organisms are mesophilic. Mesophilic, sorry, there's a typo. Okay. And if you isolate some free living organism, for example, some organism from the soil, usually the optimal temperature for those free living organism is 30 degrees. Okay, you don't want to culture them at 37, the human body temperature. They prefer a lower temperature, 30 degrees. But for most of the human pathogens, they prefer a temperature around 37. You know why? Body temperature. Why? Yeah. So that's the story of evolution. So they evolved together with their host. They have already adapted to the body temperature. So they prefer 37 degrees. Okay. And that's the reason nowadays we talk about some fungi problem, because usually the fungi live in the environment. They prefer a low temperature. Usually they don't cause infection in humans. The major reason is that our body temperature is too high, so that the fungi cannot set up infection in humans. But nowadays, because of the global warming and the extreme temperature in the environment, the fungi in the environment already evolved so that they could adapt to body temperature. So they break the temperature barrier so they can set up infection in humans. So nowadays we have more and more fungi infection in humans. That might be the reason. So okay, the third group is called thermophilic form. Do you know the meaning of thermal? Heat, right, exactly. Thermophilic means that they like heat, they like higher temperature. So they grow best at temperature between 50 to 60 degrees. And this temperature is lethal for E. coli. If you put E. coli at this temperature, they will die in like half an hour, in a minute. But those bacteria, they prefer such high temperature. If you, for example, in Zhengzhou City, there are some hot springs. If you isolate some organism from the hot spring, and probably they would like a higher temperature. So that's, again, it's a story of evolution. They have already adapted to that temperature. So if you culture at 37 degrees, they cannot grow well. And that's the first factor we are talking about, the temperature. The second one is the party is over. You guys just look like kids, not like grown-ups. It's like a baby in kindergarten. Okay, the second factor I'll talk about is the pH. pH, we learned that in high school, right? So what's the meaning of pH? It indicates the level of iron, or the hydrogen ion concentration. concentration. So the definition of P here, little p here, means minus log. So that's the definition of little p. H is a hydrogen ion concentration. And you have to write pH in this way, okay? It's important because sometimes I see students write the paper or report, they write pH with capital P, capital H. That's not correct. You have to write it in the little p, capital H, okay? So the pH indicates the level of hydrogen ion concentration. Some bacteria, they prefer pH in the middle, okay, around 7. So pH 7 we call neutral. And when they prefer pH around 7, from 6 to 8, we call them neutrophil. Okay? But some bacteria, they prefer a lower pH, such as low as pH 3. And those group, we call them acidophil. The first word, acid, right? So they prefer a low pH with acid. For example, in some mineral mine, and there's some water in it, and that pH is very low, and some bacteria can grow in it. So that's acidophil. While some bacteria, they prefer a higher pH, like pH 8, pH 9, and this we call them alkalophil. They prefer a basic condition. One famous example of human pathogen is the Vibrio cholera, which can cause serious problem in human. And they prefer a pH like pH 8 to pH 9, okay? And again, most pathogenic bacteria grow best at pH 7.2 to 7.6. Why? Right. This is the range of physical pH in human, right? So they have evolved to adapt to this pH. Like E. coli, the Salmonella, they prefer this pH. Okay, the third factor. Okay, before we talk about the third factor, there is a story of pylori. Does anyone know the story of discovery of pylori? Anyone know? No? This guy, Dr. Barry Marshall, is also a professor of Zhengzhou University. There is a center or institute in his name, starting on Helicobacter pylori. And now we know that Helicobacter pylori is a pathogen in human stomach. It can cause infection and ulcer in the stomach. And the story happened in a long time ago. That's in late 1970s or early 1980s. When he was a young doctor in the hospital, and he was interested in the reason of ulcer in the stomach. And another doctor, his advisor, told him that, well, it might be some bacterial infection, because when they scan the specimen from the stomach, they could see some brown dots, brown points. So that might be some bacteria. And they work on that. They try to isolate, to culture it. It took them a couple of years to finally culture the bacteria, Helicobacter pylori, from human stomach. It is bacteria. And after their discovery, they want to publish their discovery on some conference, some medicine conference. They submit their discovery to the conference, but it was rejected by the conference because the conference people told them that, well, they didn't believe anything could grow in human stomach because the pH there is very low, like pH 2, pH 3. And they told them that if you want to confirm this is due to bacterial infection, you should set up animal model, right? So you should follow the Koch postulates. Isolate the bacteria and then use some animal model to cause the same problem. But the problem is they don't have any animal model to use. If they infect mice or rabbits or other animal, they couldn't successfully do the animal model, set up the animal model. So, well, later, Dr. Michel realized that the best animal model was just himself. So after he isolated some bacteria from his patient, he cultured the bacteria, the pure culture, and he directly drank the bacteria suspension to cause infection in his own stomach. Okay, that was successful. But I'm not suggesting you guys to do the same thing in future, okay? The reason he could do that is that he already cured his patient with antibiotic. So he knew that some antibiotic would kill the bacteria. So after that, he took some antibiotics and cured himself. Yeah, their work was finally realized by other scientists, other doctors. But it took them another 20 years. Okay, another 20 years to receive their Nobel Prize. They got their Nobel Prize in 2005. Okay, Dr. Michel, very brave, right? Okay, he was from Australia, but he had a lot of collaboration with hospitals in China, and in the first and the fifth affiliated hospital of Zhengzhou University, some people worked on pylori, and yeah, a couple of years ago, he visited Zhengzhou University and gave a talk. A very interesting and funny guy. And yeah, he actually speaks Chinese. Not a lot, but yeah. Before his speech, he talked in Chinese, then he switched his talk in English. Very much so. Okay, the next factor we want to talk about is the oxygen. And right now, while in our air, oxygen is everywhere. Like in this classroom or on campus, you know the level of oxygen in the air. We learned that in high school. What's the concentration of oxygen in the air? 21%, exactly. So it's close to that, right? 21%. So that's the current level of oxygen in the atmosphere. But in the whole history of the Earth, you can see the concentration of oxygen changed a lot. At the very beginning, long time ago, here, okay, 1,000 million years ago, it kept for a long time at very low level, less than 5%. Then it began to accumulate, to rise, okay? And here, around 200 to 300 million years ago, the level is as high as 35%, very high. After that, it drops to about 15% and rise again and drops again. Now it's here. So have you ever asked yourself the question, what's the source of oxygen on the Earth? Why it increases and drops? What happened? What's the source of oxygen? We learned that it's a photosynthesis, right? Because of the evolution, and when there is photosynthesis, there is production of oxygen. So before photosynthesis, it stays very low. When there is photosynthesis, the green bacteria, plants, and other algae, they produce more and more oxygen. So we accumulate more and more oxygen in the air. So now we have 21%. Does that mean that all the current microorganisms like oxygen? The answer is no. Currently, if you culture the microorganisms, bacteria, some of them are obligate aerobes. Obligate aerobes means that they specifically require oxygen as a final electron receptor. So that's, remember on the cell membrane, the electron transport chain, right? And the final acceptor is oxygen molecule. So they require oxygen as a final acceptor. But there are also some bacteria, they are obligate anaerobes. Here, AI means no, right? So they don't like oxygen at all. So requiring other substances other than oxygen as a final acceptor, final electron acceptor. And they are sensitive to the presence of oxygen. So oxygen is actually toxic to them. For example, some bacteria like discovered deep in the soil, they don't like oxygen at all. If you culture it in the normal condition in the lab, they don't grow. They will be killed by oxygen. And also some are facultative anaerobes. Facultative means that, well, they are able to live in the presence of oxygen and in the absence of oxygen. So, well, they adapted both conditions. But usually, they live better, they grow better in the presence of oxygen. So these are the facultative anaerobes. And the last group, we call them microaerophile. Here, micro means small, right? So they prefer low level of oxygen. So they grow best in the presence of 5 to 6% of oxygen. And if the oxygen level is above 10%, it will suppress the bacterial growth. So this we call them the microaerophile. So it all depends. And each species have evolved ability to adapt the condition with a proper concentration of oxygen. So that is why the reason in the lab, when we culture environmental microorganisms, it's hard. Because in the environmental condition, the oxygen level might be lower than in the lab. Because in the lab, in the cabinet, usually we use the normal condition, 21%. But in the soil, the oxygen level might be much lower. So that might be one of the reasons. Okay, now we learned the nutrients requirement. And also we learned the physical factors required for the bacterial growth. Then we want to know how the bacteria grow. On the cell level, okay, one single cell level, the bacteria reproduce by binary fission. Okay, this is two words, binary fission. What's the meaning of binary? Binary means two, right? Like bi means two. So binary means two. Fission means, anyone know the meaning of fission? Split, right? So binary fission means split from 1 to 2. So that's how the bacteria reproduce. Here's an example of, okay, this is a bacterium, bacillus, okay? There is DNA in it, a circular DNA. If the bacteria need to replicate, they need to prepare 2 copies of their DNA for each daughter cell, right? So first, we need to prepare the DNA replication from 1 copy to make 2 copies, okay? Here, finally, it made 2 copies of its chromosome. After that, the bacterial cell will get longer. So we call it elongation, okay? Get longer. And when the cell gets longer, it brings the 2 copies of DNA to separate. One to the left, one to the right, okay? Then finally, in the middle of the cell, it splits. There is a fission to separate the 2 daughter cells. Finally, you will have 2 smaller daughter cells, but each carry a chromosome. And this daughter cell will grow bigger and split again. And the time for each generation, time for binary fission is called the generation time. The bacterial generation time varies, it depends, but most bacterial generation time is relatively short. Like E. coli, like Staphylococcus, they are as short as 20 to 30 minutes. So very short, right? 20 minutes, 1 cell can become 2. So within 1 hour, 1 cell would become how many? If the generation time is 20 minutes. Exactly, correct. So it grows fast and much faster than human cell. Does anyone know the generation time of human cell? If it grows all the time? No. For human cell, if we culture the human cancer cell in the lab, the generation time is about 24 hours, 1 day. So after each day, the number of human cells will double. So that's about 1 day, 24 hours. And for bacterial generation time, it can be much longer. Depends on the species. For example, if you heard the name of the Mycobacterium tuberculosis, that bacteria grows much, much slower. The generation time is about 18 or 20 hours. So it takes a long time to culture it in the lab. And usually it takes 2 to 4 weeks to see the single colony because the bacteria is growing so slowly. So this is on the cellular level, on each specific cell. And if we check the population, whole population, not focus on any single cell, the bacterial growth can be described by a growth curve. For example, if you have a tube of culture or a flask of culture, the bacterial growth can be described with a growth curve. In this growth curve, you will find that it can be further divided into 4 different phases. The first one is a lag phase. Then that's the exponential phase, or sometimes we call it log phase, L-O-G, log phase. Then it enters a stationary phase, a plateau for some time. After that, there is the decline phase or death phase. So usually in the lab, when we culture bacteria in a flask, we would get the curve. And from the curve, you can decide when you do an experiment, what time point you should sample your bacteria. For example, if you want to study the morphology of your bacteria, you want sample your bacteria at the log phase or exponential phase, because at this phase, the bacterial cell will show the typical structure, typical morphology. But if you culture it for a longer time, like in the stationary phase or death phase, the cell might die and they would show like strange structures, strange morphology. And after you stain it with color and check with microscope, they would show some atypical structure you cannot explain. But if you culture some bacteria to produce some antibiotic, you want culture it to the stationary phase. And there you can harvest more antibiotics. Okay, we'll have a break, and after that, we'll talk about each phase in detail. We're talking about the growth curve of bacteria. So it describes the whole population behavior. And let's introduce each phase in detail. We need to learn what happens in each phase. The first phase, okay, it's the lag phase. The lag phase is here, the early point. After you inoculate some bacteria to some new medium, and you monitor the increase of viable cell continuously, you would notice that for first several hours, the increase is slow. So we call it lag phase. It's slow. It doesn't mean that it's quiet inside the cell. So the lag phase represents a period during which the cell adapts to the new medium, new environment. And also, inside the cell, it prepares enzymes and other molecules, other intermediates, which are required for the growth, for the replication. So even though the cell number is not increasing dramatically at this time, it's still producing a lot of molecules for replication. So when the enzyme and intermediates are ready, the concentration is high enough, the cell will enter a new phase, begin splitting, begin the binary fission. So that's the exponential phase or log phase. The log phase, okay, the second part, that's the cell number increases in an exponential way. Or you can calculate the slope of this part. And this continues, okay, continues until something happens. Either one or more nutrients get limited, get exhausted, okay? When the cell grows, it utilizes the material. When the nutrients get exhausted, well, it stops increasing. Or if the bacteria produce some toxic molecule, toxic metabolites, the toxic metabolites will accumulate in the tube. If the toxic metabolite product gets high concentration to some level, it will stop the bacterial replication. So this is exponential phase. Then it enters the third phase, the stationary phase, or sometimes we call it the plateau phase. In the stationary phase, the exhaustion of nutrients or the accumulation of toxic products causes the growth to cease completely. So for some time, some period here, there is no increase of viable cell. If you count the viable cell number, it will keep stable for some period of time, this stationary phase. But after that, it will enter another phase, or death phase. The viable cell number will decrease, or sometimes we call it decline phase. In the decline phase, the death rate increases until it reaches a steady level. Okay? And frequently, after the majority of cells have died, the death rate decreases drastically here. Okay? So that's the death phase. And this is a very traditional method to describe the bacteria's behavior when you culture it. But it's important. And I did this when I was in graduate school. And now in my lab, my students, sometimes they need to do this experiment in the lab. And you can see the time from 0 to 40 hours. So which means that when you derive the growth curve, you need to stick to the experiment for a couple of days, right? You need to sample the culture every several hours, every like a couple of hours, measure the OD or measure the live cell number. So you have to sometimes sleep in the lab. You stay there overnight to collect more data. So to get this curve. But nowadays, with some new equipment, some machine, you can leave your sample, the culture in the machine, and the machine would collect data every hour, every 2 hours. So you just leave the sample, leave the culture in the machine, and you don't have to stay in the lab for a long time. So that's a growth curve. And yeah, now we learn how the bacteria grow, how the population grow. So one goal of this is that, well, we can culture bacteria in the lab. So that's important. Like in the future, when you are a doctor, you collect the specimen from the infection site, and then you isolate the pure culture. Well, it's some bacteria, and you want culture more bacteria of this isolation, so that you can study the property. And you want to know the antibiotic resistance, or sensitivity, so that you can pick the proper medicine for your patient to cure them. That relies on cultivation. So this is important to culture bacteria in the lab. And we use culture medium to culture bacteria. This is the artificially made mixture containing the essential nutrients so that we can culture bacteria in it. And based on their function, the culture media can be further divided into several different types, like basal medium. So that's a basic medium which can support the bacterial growth. Or we have enrichment medium, selective medium, differential medium, and also anaerobic medium. Let me show you each type in detail. The first one is enrichment medium. This is the medium. It contains some chemical in it, some component in it, supporting the growth of some specific type of bacteria. However, the enrichment medium may have some selective feature. So probably your sample is a mixture containing different type of bacteria, but this enrichment medium can allow certain bacteria to grow better than others. So that's enrichment. So it looks like it has some selective feature. For selective medium, it contains certain chemical which can inhibit the growth of certain types of bacteria and supporting the growth of desired species. So kind of similar, right? So this one is selective, like this one. This medium is called McConkey medium. It's popularly used in hospitals. In this McConkey medium, it contains some chemical which allows the growth of gram-negative bacteria, the bacteria with outer membrane. But gram-positive bacteria cannot grow on it. Okay, so this is the one of the selective medium. It selects the gram-negative bacteria to grow, while kill the gram-positive bacteria. And similarly, McConkey medium can also act as differential medium. Differential medium means that it causes the colonies of a particular type of organism to have a different appearance. This is again McConkey medium. And if your bacteria can utilize lactose, that's the sugar already added to the medium. If it can utilize lactose to produce acid, the pH will drop. When the pH drops, in the medium, it contains pH indicator, so the color will change to reddish color. So the red color means that there is acid produced, so that this bacteria is able to utilize lactose to produce acid. While on this half, the bacteria cannot utilize lactose. No acid production, so the color is still yellowish color. So McConkey medium can also work as a differential medium. You can also design anaerobic medium, and this medium is used for the culture of the obligate anaerobes. Okay, for these obligate anaerobes, they cannot utilize oxygen as the final electron acceptor. So that means that in the medium, you need to add in some chemical as a final electron acceptor. Some type of chemical, some type of salt in it. Okay? So this picture shows us the difference of selective media and differential media. Selective media means that it selects certain type of bacteria to grow, stand out on the surface. While this differential medium means that different bacteria would show different appearance when it grows. So you can tell the difference according to their either their color, the texture, or the pH, like the pH indicator. So, but just keep in mind that any specific medium can be both selective and differential. It all depends on its function and how you use it. Okay, if we classify the medium according to their physical state, there are three types: either solid medium, liquid medium, and semi-solid medium. Okay, liquid medium, it's easy to understand. It's liquid, so it flows, and we don't add any agar or any solidifying component in it, and it supports the bacterial proliferation. But in the liquid medium, the bacteria can grow in different ways. If the bacteria can get into the liquid, it can form this suspension, bacterial suspension, to make it turbidity. So this, like E. coli and Staphylococcus, they grow this way. So this is one type. Another type is, okay, this is a liquid medium, but the bacteria grow here, the particle here. It grows as the air and lipid interface, just on the surface. So that's another type. or the bacteria might precipitate to the bottom and grow in this sediment. It all depends on the type of bacteria. So biology is full of diversity and a lot of interesting things. I don't know whether they will show you this or not this Friday when you go to lab. Do you go to the lab today, this Friday? Or next Friday? Next Friday, right? This Friday? Oh, you have different labs. Yeah. Probably you will see these tubes prepared for you. Okay. A second type of medium is the solid medium. Solid medium. When we introduced Dr. Robert Koch, the German guy, and we mentioned that it was in their lab who invented the solid medium. They added the agar component to the medium. When the medium temperature is high, the agar will dissolve into the medium. Then, when it cools down, the agar will solidify again so that the medium looks like solid. Even though it's solid, but over 98% of the medium is still water, so that the bacteria can grow on the surface. And with this solid medium, we can strict bacteria like this, so that finally we can get this single colonies. So this is useful for isolation of pure culture. So this is the experiment you want to do for the first microbiology lab. You would strict the plate like this. And finally, if you are successful, you would get single colonies, two different type of single colonies, so that you can get the pure culture. This is a common technique. We use it every day in the lab. But over 100 years ago, this technique is kind of revolutionary because you can get pure culture from the sample. And if you culture it in the liquid, you will never get a pure culture, right? So this is very important by Dr. Robert Koch's lab. Okay. And another term we want to understand is bacterial colony. That's the visible cluster of bacteria growing on the surface or within a solid medium. And bacteria within one single colony, okay, is a descendant of a single ancestor cell. So each single colony is derived from one single ancestor cell. So it's pure. All the cells within the single colony should be the same, okay? And based on the texture of the colony type can be divided into like smooth colony, like this. You can see the shining on the surface, or it can be rough, like this. It's a rough colony. Or the third one, that's mucoid. Mucoid colony, like this one. It's also smooth, but the mucoid means that this colony can flow, okay? It can spread. So when the bacteria grow on the surface, it can spread on the surface. So we call it mucoid. So that's three different type of colonies. If you culture E. coli, it should be like this. Okay, the last type of medium is the semi-colon medium. Semi-colon medium means that when you prepare the medium, you add less concentration of agar, so that the medium is between like solid and liquid. It will solidify, it does not flow, but it's very soft. Because the agar concentration is low, there should be some big holes between the agar molecules, so that when you culture the bacteria in this semi-solid medium, if they can swim, they can still swim in this semi-solid medium. So this is useful to study the bacterial motility. What we do is that we make the semi-solid medium in this tube. Then we use a sterile needle, a straight needle, to inoculate bacteria in the middle in a line. It's like this line. Use a needle to puncture a hole, a vertical hole in the middle. If the bacteria cannot swim, they will only replicate in the line. Okay, this is the bacteria without flagella. But if the bacteria have flagella and they can swim, they would spread from the line to the surrounding medium. Okay, like this one. So this bacteria is mobile. So we can study the bacterial motility with this semi-solid medium. Very useful medium in the lab. And when we culture the bacteria, and they are able to produce a lot of type of molecules, like they are able to produce pyrogen. So that's the molecule which can cause fever. And they are able to produce different type of toxins, including both endotoxin and exotoxin. We'll talk about this in detail in future. And some of them are able to produce antibiotics. And nowadays, the antibiotics we are using, most of them are from either bacteria or fungi, such as polymyxin B or other polymyxin, or this one, bacitracin, I don't know the Chinese name, and also the streptomycin, and those are all produced by bacteria. And they are able to produce bactericin. It's a molecule which can inhibit other bacteria, but it's different from antibiotics. And some bacteria are able to produce vitamins, such as vitamin B and vitamin K. And in our digestive tract, in the intestine, some bacteria are able to produce vitamin B molecules and vitamin K. And it's important for our health. Or some bacteria can produce molecules with color, pigments, such as the pigments can be fat soluble or water soluble. Pigments can be different color, black, like red color, yellow color. Here are some examples. Famous example of the Staphylococcus aureus, a very important human pathogen. They are able to produce some yellow or golden pigment. But this pigment is not water soluble, it's fat soluble. So when the pigment is produced, it's associated with the cell membrane. It does not diffuse into the medium, into the agar. So you could see this brown, this yellow or golden colonies. That's because of the production of pigment. Another example is the water-soluble pigment produced by Pseudomonas aeruginosa. This pigment is a greenish color, but it's water-soluble. Water-soluble means that it can diffuse in the medium. And when you culture the bacteria, the bacteria would produce a lot of pigment, and that pigment would stain the whole plate into green color. Another interesting thing about this Pseudomonas aeruginosa is that when you culture this bacteria in the cabinet, in the lab, it smells good. It smells like fruit or flower. And some people say that it smells like grape juice. So if you visit some lab, open their incubator, and you smell some grape smell, you know that they are culturing this bacteria. So I have it in my lab. If you want, you can come to my lab and see this. Pseudomonas aeruginosa. Okay, this is for the first part.